Hi everyone, thank you for being here. We have the pleasure to host today Catia Pesquita. I had the pleasure to meet her this morning also and talk with her. She's an amazing professor. She's an associate professor at the Faculty of Sciences in the University of Lisbon and vice director of LASIGE. Is that correctly spelled? Where she coordinates the research line in excellence in health and biomedical informatics. So good place to be right now. She is passionate about exploring the synergies between human and machine intelligence in generating scientific knowledge and has made significant contributions to knowledge graph and AI for science. So I think her talk will be amazing today. I have very good expectations, so please give a warm welcome to Catia and let's enjoy the talk.
Thank you so much for the very nice introduction.
Okay.
I was thinking of standing here instead of behind the table because I'm very short. Is this okay for technical purposes? Can I stand here? Perfect. Thank you so much. So thank you for the warm welcome to Barcelona Supercomputing Center. Although I've been many times to Barcelona, this is my first time here. And so far I've had an amazing morning with Claudia, Victor, and Gonzalo and his team, talking a lot about trustworthy AI and a lot of proteins as well. We'll get to the proteins later in my talk. So today, let me see if I can change my, they're saying they cannot see the slides. Can somebody help me here? And also, I'll see if I can change them. On Zoom, they're saying they cannot see the slides. I haven't changed my slides yet. I'm trying to change them. I'm not able to change them with this.
You should. It was working before.
The universe wants me to stand behind the table.
No.
I don't know. No, just give us a second and we will start. Let me see if I can change here. If this is... Okay, here they can change. Can you try again? Yeah, there was just stuck. Yeah, okay. Technical issues aside, let us continue. So the title of my talk has caused some confusion this morning. I was asked if I was a neuroscientist. I am not a neuroscientist. The neuro stands for neural networks. Okay, I was originally trained as a biologist, like many of you, and then I kind of morphed into a computer scientist along the way. And today I wanted to talk to you about a different kind of AI, neurosymbolic AI, and why I think it is a really cool paradigm in AI to solve AI for science, and in particular to support explainable scientific discovery. I will not be very technical in my talk, but your questions may be as technical as you want them to be. This talk will be a little bit more to share with you some ideas and challenge you to think more deeply about some of the most important questions in AI for science. And with all things, we start in ancient Greece, when all sciences were one science, and we did not have these artificial divisions between biology and physics and philosophy and logics. And people like Aristotle worked on all of them at the same time, and how that lack of formal boundaries between sciences also brought about some very interesting and fast developments. So if we go back to Aristotle's categories and his first works into logics, they all are born from also his desire to understand the natural world and to categorize every living thing. Of course, Galen and his many endeavors into the clinical side of things also brought about the first developments in modal logics. And of course, this was not limited to ancient Greece and Rome, but also Middle East and Arabic researchers if there was such a thing back then, like Ibn Sina, whom perhaps you know more from the Latin name Avicenna, also worked at the intersection of logics, mathematics, and life sciences. So let's jump forward into the 20th century, where life sciences were actually two of the first proving grounds for the first real-world working AI systems. Both Dendrol and Mycin, both developed in the 1970s, were really important things for classical AI. So the AI that is based on knowledge systems that try to encode knowledge in sets of well-established rules that are built based on expert knowledge. And Mycin was used to test scientific hypotheses about the properties of molecules, and Dendrol for predicting a type of infectious disease from culture data. But these systems were never put into production. They proved to be too brittle. There was a lot of knowledge necessary to interpret this complex phenomenon that could not easily be captured by rules extracted from domain experts' knowledge. So let's jump again 50 years into the future, into this loud revolution of AI in science, where machine learning is enabling us to predict accurate protein structures just from amino acid sequences, to produce millions of new materials, and where papers that mention AI on the title or on the abstract are increasing not just in computer science, but across all sciences, and poised to reach perhaps maybe 10% of the papers published this year will have AI on the title or the abstract. But at the same time that we've all been witnessing this very loud revolution of what we can do with statistical learning and figuring out the patterns that govern natural phenomena and pushing forward scientific discovery, there has also been another revolution going on, more or less at the same time, the last 20 to 25 years, which I like to call the quiet revolution, the revolution that you perhaps use every day, and many of the people with whom I talked to this morning already also use it, but perhaps don't think of it as a revolution. And this is the quiet revolution in AI for science. It's the amount of scientific knowledge that is being encoded in ontologies and knowledge graphs across all areas of human knowledge. Here we have a representation of the linked open data cloud that connects all of these data resources that are described with scientific context attached. And I would like to challenge you, can you read the label? It's more interesting if you can't. So if you can't, this question is more interesting. If you can't read the label, could you guess which color is life sciences on this image? Can you speak up? Red. So the light reddish color where the background is really dark that you see there, that's life sciences. And you know why the background is darker? Because every link between different data set in the linked open data cloud is represented by an edge in this graph. And the many, many edges that connect the many, many data sets in life sciences turn that background dark. That means there's not just a lot of data imbued with knowledge, data that is connected to ontologies and described in knowledge graphs, but also that this data is going across domains, connecting different perspectives to think about life sciences in different perspectives. So this brings us to the new age that we are living, where science coexists with AI, where I think everybody that I talked to today is either already very much doing AI or thinking about doing more and more AI, doing more machine learning, exploring more of these opportunities. But what does this actually mean for scientific discovery? Are we using AI as just a tool to produce more data, just to facilitate data analytics, or are we actually using AI to do actual science? And so I think that there are two major challenges in AI for science, in turning AI into a true epistemic tool, something that helps researchers into creating actually new knowledge, into understanding the natural phenomena that they are studying. And these two major challenges, I think, are the integration of domain knowledge. How can we take what we already know, the knowledge that science has accumulated decades upon decades, all of the efforts in big data that we have seen in the last 2 decades of building huge data infrastructures for science, how can we benefit from that domain knowledge and use it in these AI tools? There is this tendency to think that scalability is enough, that given enough data, you can learn all the patterns from data that is as raw as possible. Just this morning, we were discussing AlphaFold and its ability to learn protein structure from sequence data alone. But if you really think about how AlphaFold works, you will figure out that there is some knowledge that went into building the architecture itself, and there is some knowledge about that conserved sequences mean a shared evolutionary path and how important that is to actually learn the structure. And so in some ways, the way that scientists build that architecture is informed by domain knowledge. Although it's fed with, of course, a lot of sequence data, there was some domain knowledge there. And there is a lot more domain knowledge out there produced by this quiet revolution that is just begging to be integrated into the AI models. And the other challenge is explainability. And this is perhaps an even more important challenge. And this kind of feeds into my particular view that science is a human endeavor. Science without scientists, science without humans to understand things is not really science. It can help science. It can propel scientific discovery. It can benefit humanity. But the ultimate purpose of science is understanding by humans. So I like to quote Carl Sagan on this, that we are a way for the cosmos to know itself. And if we want AI to really be a tool for scientific discovery in the sense that it helps humans make sense of our world and our universe, then explainability must be an integral part of this. Of course, explainability is also very important for trusting, for auditing, for correcting, for debiasing, for all of those other areas. But fundamentally, for scientific discovery, it's a tool of interaction between AI and humans in this co-creation of scientific knowledge. I'm sorry, okay. Okay. Which brings us to the first topic in my lecture, which is what the hell is neurosymbolic AI? It's not neuroscience. It is actually a field of AI that tries to combine, to marry two long-standing enemies in the field of AI, the symbolists and the connectionists. And I will tell you a little bit of the history of this so you can understand why this is a little bit controversial in a sense. So if you remember when we were discussing the beginnings of AI and the symbolic systems that were based on rules but were too brittle to really be adopted in the real world, those were the systems that come from the symbolic part, the part where you capture the knowledge, formalize the knowledge out there in a structured way that computers can interpret, and then run reasoning over those sets of rules. And then the connectionists are the side of AI that runs on a different paradigm, that runs on the machine learning paradigm where you show data to a model, the model can learn to abstract the inherent patterns on the data to figure out some interesting aspects over that data, and that everything there is to be learned can be learned just by looking at the raw data. You just need more of it and more of it, which is perhaps the paradigm that we are now seeing, of course, behind the loud revolution in AI, the machine learning, deep learning, neural network revolution of AI for science and AI elsewhere. In neurosymbolic, the paradigm is why not get the best of both worlds? If we have a way to structure what we know, the symbolic part, but also a way to learn from the data that we are collecting, why not try to get the best of both worlds? The truth is that many have tried and many have failed, and this is not an easy paradigm to work on. It brings a set of technical and methodological challenges, and it has been only in recent years that neurosymbolic AI has really been gaining some traction due to two things. First, improvement in the neural part with the development of deep learning and better neural networks and the computational capabilities afforded by high computing performance and GPUs, but also the increasing availability of symbolic models that capture more and more of our knowledge, and of which the ontologies and knowledge graphs in the life sciences that I showed you before are a very important part. And so we have these two basic ways to look at learning. On the top, data goes in to train a model that learns to deduce a symbol, a label. If you think of this as a model for supervised learning, you can learn characteristics about your data by training this model to separate the search space according to those characteristics. In the bottom, you have a different perspective, the symbolic perspective, where symbols put together, they converge into a model that represents the semantics, the meaning of a given representation of the world, and then you can use reasoning algorithms to deduce new facts. So for instance, Catia is from Lisbon. Lisbon is in Portugal, therefore Catia is from Portugal. And so these are two very different ways, and the goal of neurosymbolic is to combine these two strategies to arrive at different architectures that can get the best of both worlds. And here I have a very simple example of one where the model takes as input symbols. It can derive new symbols by deducing over the existing symbols. So based on pre-existing rules, derive new rules, or based on pre-existing facts, derive new facts. Catia is from Portugal. Catia is from Lisbon. Lisbon is in Portugal. Deduce the new fact that I am from Portugal. And then use those together with data to train a model. And therein lies the answer to one of the first challenges that I told you about, how to integrate prior knowledge. If you have your prior knowledge encoded into this symbolic layer, and then you can show the symbols to a model that can both learn from the symbols and the data, then we can have a true neurosymbolic system, a system that learns from both data and symbols at the same time. But it can also be used in a different way to solve the second challenge, the explainability challenge. And in here, you will find that data is used to train a model and to deduce a new fact about the world, a new symbol. For instance, that we were discussing protein function this morning, okay, that a protein for which we do not know its function, now we infer that it has a specific function, let's call it ion binding, and then we can take that statement, that fact, represented as a symbol, and check it against the existing knowledge, the semantic model that we have, in this case of protein function. Does it make sense with what we know about proteins that this protein may have that function? And so in a sense, this allows you not just to validate your prediction against the domain knowledge, but also to arrive at an explanation. Does this function assignment make sense for my protein? So these are kind of like the two basic architectures for neurosymbolic AI that are perhaps a background necessary for the rest of my talk. But let's go back to where that semantic model or where that knowledge comes from. I work a lot with knowledge graphs. Knowledge graphs allow you to have the ability to describe entities in the real world. And since I do a lot of work in life sciences and in healthcare, for me, those are biomedical entities. And they allow you to describe these entities across multiple different perspectives in an integrated way with meaning that can be interpreted by algorithms. So for instance, here, we can represent a gene through which compounds it binds to, which diseases it is associated to, which molecular functions it performs, where in the cell is it active, if it binds to a compound, what are the side effects that can be caused by that? So you have this network that represents the entities with specific definitions for anything. And so knowledge graphs, in a sense, are explainable by design, and they give us the answer to how to integrate prior knowledge, they encode that prior knowledge, and how to afford explainability. They are explainable by design, although they don't look like that when you load them into memory. This is their conceptual model, so it is fully interpretable. However, most of the machine learning algorithms that work over knowledge graphs, to integrate this prior knowledge, they sacrifice that interpretability. They sacrifice the explainability. Why? Computational convenience. It is much easier to work with numerical vectors. And so in a sense, all of the algorithms from knowledge graph embeddings to graph neural networks, they sacrifice that explainability for computational utility or convenience, let's say it, based on the assumption that, okay, it's okay if we lose the explainability as long as we retain the performance. But if we go back to our two desirable properties in AI for science, are we getting prior knowledge integration? Yes, we are. Okay, the vectors that are transformed from knowledge graphs still retain that knowledge in a way, but do we have explainability? No, we have sacrificed explainability. And so we have been doing a lot of work to revert this and to show that, yes, you require good performance, good accuracy for any output in AI for science to be valuable. You don't want to sacrifice that. So the question becomes, can you still have good enough performance without sacrificing explainability? But what do we actually mean by explainability? And this is a big question. This is where we go a little bit philosophical in my talk, and I promise you by the end we'll get back to the algorithms. So in my perspective, the goal of science is explanation. So instead of saying that one of the goals of science is explanation, we could simply say that science is explanation. It's just us trying to make sense of everything around us. And there can be no explanation without semantics. So this is a favorite quote from my friend Giancarlo. And what does this mean is that if you do not have a shared meaning with others, if you do not have a common ground to stand on, you cannot explain anything to anyone. So the explanation is a co-construction of the knowledge. It requires not just the communication between scientists and researchers, something you do every day, but also if we are still trying to achieve this goal of AI for science, we want to support a communication between whatever the models are figuring out and our own understanding of what they are doing. But this brings us to a very problematic question. A question that I think nobody has a good answer for yet. A question that I would wish would be true because it would make some of our endeavors easier. And the question is, do we have a fundamental relation between the explanation of a natural phenomena, how something happens in the real world, and the explanation of the output of an algorithm that tries to understand that natural phenomena?
Oh.
So,
No, estoy limpiando mi home directory, pero me gusta hacerlo.
I don't know. Was that a question for me? I'm not sure. I think it's someone online and maybe they're not muted, but if it was a question, I will be happy to take it. Okay, okay. I don't know. There could be a question. This is very controversial, right? so, so let us dig deeper here. So the question is, if we train a model to predict whether two proteins interact, and we figure out an explanation for how that model arrived at that prediction, does that explanation have anything to do with why the two proteins interact in the real world? That is the question. If yes, then we can use AI to actually query and investigate and research the real world phenomena. If not, if it's just a spurious correlation, nothing to do with a natural phenomena, then we are lost, we are doomed, and I can retire. So, answering this question, if this fundamental relationship exists, for me is the fundamental piece in the puzzle of whether AI can be a real tool for scientific discovery or not. But this brings us to two very different interpretations of scientific explanation. Philosophers of science are worried on defining it. What is a scientific explanation? And in the last 5 years, I've read many philosophy papers on the topic. I still cannot give you a precise definition, and I think it will be continuing to change as our ideas on science also change. But luckily, AI scientists are a little bit more pragmatic, and they are less concerned with defining the scientific explanation and more concerned with generating the explanation, right? That's what you see in all of those papers on explainable AI. How do we generate explanations? Curiously enough, it's been only recently that people are more worried on how do we evaluate an explanation. For the longest time, just presenting a list of important features was considered good enough. It is not good enough. It is not nearly good enough. So what is an explanation? And for this, I would like to give you with a simple model to think of explanation. An explanation is an answer to a why question. Why does copper turn green when exposed to air? And if we think about it, there are many components to an answer to this question. Copper turns green when exposed to air. It's the pair that connects the why question and the presumption. So the why question, why does it turn green? I'm sorry. And the presumption, due to the formation of a protective patina, etc., etc., etc. And here you have a connection between this phenomena and some explanation of to why it happens. Of course that this is a very simplistic view. This does not take us into more complex questions on to whom am I answering this why question. Different people would require different types of explanation to be satisfied to learn why copper turns green when exposed to air. I have a question. Yeah. That is a very interesting question, the why and the how. Okay, so if you think of why in terms of volition, in terms of a motive, do we have a motive for copper turning green when exposed to air? So there is some, I would say, confusion between the how and the why in explaining some phenomena where there is no volition, so there is no actor deciding anything. If you are explaining the actions of a person, it is quite different if you consider the why and the how, right? So, you know, why does the person cross the street is different from how does the person cross the street. But if you remove that component, it is very common that the how/why questions may be answered in very much the same way, right? So another way to think about explanation is to think about causality and to think about counterfactuals. So there are there is a big current in explainable AI that tells you that the only explanation possible or the only valid explanation is a causal explanation. I do not necessarily adhere to that. I think that we may have different levels of explanation that are still useful, although they do not give you a causal relationship. But in counterfactuals, they give you the ability to ask a what-if question. What if this property of my system changed? How would that affect my prediction? This morning, we were discussing something that was a little bit counterfactual in a sense. What if I change this amino acid in my protein domain? What would happen to the protein function, right? That is a counterfactual question. And it allows you to what? To explain the role behind that change in the involvement on the phenomena. So what is the role of this amino acid in ensuring that this domain performs this function? And of course, you can take this further to causal chains and chains of events that influence each other. Another way to think about explanation are necessary and sufficient causes. Here in necessary and sufficient causes, we can think of the necessary causes as all the events that are necessary to cause the event in question. And a sufficient cause is something that is able to cause the event only by itself. Please keep necessary and sufficient in mind because this will be needed further along the line today. And I really like this model of scientific explanation by James Overton. So James Overton is a philosopher turned bioinformatician. So there are many ways to become a bioinformatician. And James thinks of this model that goes from data to theories, that a theory can justify a model models all the kinds of things in it. The kinds can be instantiated by real-world entities, and these entities can be measured by data. And all along of these steps, you will see that there are opportunities for pitfalls. Let's start with data. The way that we measure something can introduce a bias. We tend to think that, okay, data never lies, data is all truthful, humans just introduce bias, but we introduce bias in the data just by selecting the types of measurements and instruments and samples that we are going to use. So that's number one, right at the first step. Then, how do we describe the data? What are the entities that we choose to aggregate our data on? Are we going to look at this data from the perspective of individuals, from the perspective of genes? If I analyze the same data set focusing on genes or focusing on the people that have those genes, I will arrive at different conclusions, and that has happened in my lab before. So the entities that you choose to assign to represent the data also bring a set of challenges. And this continues into the concepts or the kinds and how you decide to model these concepts into a more complex model, and finally to arrive at a theory. All of these steps build explanations. You can have an explanation from data to entities, kinds and models and theories, and it is very, very rare that you arrive at the final stage. A lot of the things get lost along the way. But since we are interested in generating explanations, because we are AI scientists, we need to understand, okay, if we are going to generate an explanation, what properties does that explanation require? And in our work, we have been using this framework for scientific explanation proposed by Michael Kias, where explanations require, or explanations should have properties that fall under 4 virtues. Evidential virtues, which are concerned with an explanation being accurate to what it is trying to explain, providing causality, explanatory depth. coherential virtues where the explanation doesn't contradict well-established knowledge and doesn't contradict itself. There are cases of explanations in explainable AI that are actually illogical and inconsistent. Also, aesthetic virtues, which are a little bit of a harder sell for the AI specialist, such as beauty, simplicity, and unification. But you can also think of this in a cognitive perspective. What is the minimum amount of information that you need to give your scientist about the explanation for the scientist to have a complete understanding? Because if you also show too much, you will overload them cognitively, and they will probably not understand the explanation. And finally, perhaps one of the most important virtue and the one that is ignored by most explainable AI works are the diachronic virtues. So, does your explanation lead to new knowledge? Does your explanation stand the test of time? If by looking at an explanation, if I get to ask a new question, to pose a new hypothesis, to design a new study, it will be fruitful and it will be applicable somewhere else. It will not just give me a trust on that a specific AI output that I'm looking at, but it will contribute to further scientific discovery. So the question, how can we generate explanations? And everything starts with explanations of what? We spent the morning discussing protein-protein interactions, and I promised I would talk a little bit about protein-protein interactions, and here they are. And the question is, why do these two proteins interact? But if you remember the definition of explanation as an answer to a why question, would lead you to think, okay, is this really the question that I want to ask? Or is this really the question that I can answer? Because actually, the question that we were asking, although related to protein-protein interactions, was not why do these proteins interact, but rather, why are these two proteins similar? Our hope, of course, is that if proteins are similar, they could be more likely to interact, but the real question was not why they interact. This work was not actually answering that question. This was the real question that we were asking. Why are these two proteins similar? And a classical way to measure the similarity of proteins using ontologies and knowledge graphs, if you remember that's where I started my research, is using semantic similarity. So algorithms that look at the properties of the graph composed by the ontology and measure the similarity of entities in this graph. Here we have a graph, which is actually the gene ontology, which I am sure many of you are familiar with. And the two proteins, P1 and P2, are described with gene ontology terms, making this nice subgraph. And we measure properties of this subgraph to determine a single numerical score that measures their similarity. If we are lucky, we can get 3 similarity scores because the gene ontology is divided into 3 branches: biological process, molecular function, and cellular component. So, is it interpretable? Well, in a way, you can say, okay, these two proteins are 90% similar in the biological processes that they perform. You could look at the annotations and you could see them, but it is also very not fine-grained, right? We are looking at only these 3 scores of these 3 very broad properties, and it's not really giving us an understanding of why they are similar, right? We cannot say, okay, they are similar because they perform 90% similar biological processes. Is that sufficient? I don't think it's quite interesting. So our question was, can we make this more relevant? Can we have some more explanatory depth into this system? And so we proposed a method to generate vector representations of protein pairs that are based on different ways to measure their similarity according to different branches of the gene ontology. And then we use these vectors to train machine learning models to predict protein-protein interaction. And now we are using the similarity of proteins as a representation of their features to learn the interaction. We just call it KGSIM to VEC because it's knowledge graph similarity to VEC, into a vector. And what we did was generate explainable features, and these features are still a similarity score, but now instead of being very high level, biological process, molecular function, we smartly designed subgraphs that represent more relevant aspects within the gene ontology hierarchy for those two proteins. And now we can say that these two proteins are similar because they both do ferric ion binding, or because they both do oxygen transport. And then we used supervised learning over these vectors of similarities, but now each of these dimensions is interpretable. When I started my talk, I told you, okay, we learn these vector embeddings and we lose the explainability. It's because each of those dimensions in those vectors is no longer associated with a specific portion of the knowledge graph. But our vectors are. We know exactly what each dimension captures, and it captures specific aspects of protein features, functions, biological processes, and cellular components. And so what that allows us to do is to not only predict the interaction, but also to generate an explanation. And our explanations look like rules. For generating these explanations, we used either models that are fully interpretable, like decision trees or genetic programming, and those models generate rules from the start, or we used explainability algorithms like LIME and LORE to generate rules over black box algorithms. And now you have explanations for why the two proteins are similar and therefore interact. And the explanations look like, if they have high cellular and anatomical entity similarity and they participate in a very similar metabolic process, then they're likely to interact. And now you have more specific rules that capture why they are similar and why they are likely to interact. In the end, to show to a user, the explanations look like this. They look like a series of semantic similarity between the two proteins that we are trying to explain. And this is a quite interesting case. So this is a false positive case in our data set. So these two proteins had no known interaction, and they were used as a negative example for our test set. But the explanation was actually quite plausible, and the strength of the prediction was high. And so we were interested in seeing where that would take us. But first, before understanding that, remember that I told you that it is very good that we have explainability, but we cannot sacrifice performance for explainability. Because if you lose performance, then you're defeating the purpose, right? You want to be able to keep performance, but improve explainability. And here what we actually demonstrated is that in most cases, we need not just keep performance, we actually improved performance. Having this more specific and knowledge-imbued representation actually resulted in a better performance in protein-protein interaction. And if we combine different models, we even achieved better performance. In the end, why do these two proteins interact can be answered not just because they are similar, but also by asking a counterfactual question. Would these two proteins interact if they did not share a given function? So by reframing the question, we reframe also the predictive and the explanatory purposes of our task. If in the first work we were interested in understanding the similarities, And that is a type of explanation, understanding the similarity between two entities. Here, we go a little bit deeper and we go into the counterfactual explanation, the what if question. What if these two proteins did not share this function? Would they still interact? And here, we have still the same goal. We still have two entities represented in a knowledge graph. We're still going to train a classifier to predict a given relation between our entities. In the case of proteins, it's if they interact. And in this work, we were generating a different type of representation. Here, we generate pair representations. In the previous work, we generated based on similarity. Here we generate based on the shared representations. We start by looking at a graph where we have our two entities, in the case two proteins, but this has been tested in other knowledge graphs, also for gene disease association prediction. And here we have two proteins described in a graph, and we extract the common ancestors that they have, the most specific common ancestors that they have, and generate embeddings that represent those shared functions. You see how we are going with this, right? The question is, would they still interact if they did not share a function? So we start by representing the shared functions. We train a model that aggregates all of those shared functions into a global pair representation. And then we ask our what if question. We ask, if we remove from our representation one of these shared functions, does the model still classify my protein pair as interacting or not? If by removing one shared function, the model flips its prediction, it stops saying that they interact, that shared function was a necessary explanation for that interaction, right? Because by removing it, we stop being able to make the original prediction. In another way, if we have all of the shared functions and we just leave one to be the representation for the prediction, and we can still make the same prediction, that function is a sufficient explanation for the process, because it is sufficient that the two proteins share that function for the model to still give us a positive prediction on the interaction. And so by doing these removals of different aspects, we can understand the role of each different shared function behind that interaction. And again, without sacrificing performance. So we were able to improve against all baselines in protein-protein interaction prediction, and in nearly all of them in gene disease association prediction, and it was very close for RDF2-VAC. And we measured our explanation effectiveness based on how much removing the necessary components or using just the sufficient components affected the performance of the metrics. And what we demonstrated is that our methodology generates both sufficient and necessary explanations that are effective. And we went a little bit further because the same protein pair was predicted by the previous study and this study as being a false positive. And we had very good explanations for it actually being a true positive. So we selected a few examples, 4 of them actually, because this was a favor by the biochem lab in my university, and produced the experiments to detect the strong interaction between those two proteins. 2 of them were never possible to run, and out of the other 2, one was inconclusive, maybe because the PhD student wasn't doing their best, I don't know. But the other one was proved true. So it kind of shares or sheds a little bit of light into how something that was unknown so far And by looking at that explanation, we can now produce new knowledge because we generated a hypothesis. Is this a real interaction or not? And it was validated in the lab. So it kind of demonstrates the role of explanation, not just in giving you the trust on that outcome, but also in highlighting gaps in our existing knowledge. And then there's, I still have, I will go faster, I promise. We're running out of time. And then we also have another work that I would like to share with you based on drug repurposing. So the question of why is this drug a promising candidate to treat that disease can be reframed at what mechanisms can explain this drug's effect on that disease. A more specific question, a question to which we can attribute some degree of explainability. And here we have an example of an explanation extracted from a knowledge graph that is based on a path that connects the two sides of a hypothesis. So how we're starting hypothesis is eplerenone can treat hypertension. This link is not in the knowledge graph, but we find a path in the knowledge graph that could help us understand why eplerenone could be repurposed for hypertension. In this case, we have some causal detail because eplerenone is an aldosterone antagonist, which includes a drug, spironolactone, which also treats hypertension. Here on the top is another explanation that can also give you some level of causal detail. Eplerenone is a drug, drugs treat diseases, and hypertension is a disease, but it is devoid of relevance. There's no scientific relevance there. Although for a pathfinding algorithm, both are equally valid. Both have the same size, and so both connect the two ends of the hypotheses, but one of them is scientifically relevant and the other is not. So the challenge is, how do you distinguish between them? And the other property that we covered earlier was completeness. Can we give an explanation that is not just causal adequate, relevant, but also complete? In this case, there can be multiple paths in the knowledge graph that are scientifically relevant and that help you get a better understanding of why this drug could be repurposed. And finally, can we have some degree of universal coherence in our explanation if we enrich this explanation with the kinds of things in the explanation? Remember, from the scientific explanation model, this was just based on entities, and now we add a next level of kinds, and in here we just add the types of drugs that we have and other properties at the ontological level. But we also have the aesthetic virtues, and simplicity is one of them. And simplicity comes at a trade-off with the others. If you want to have coherence, you need to add information about coherence. If you want to have completeness, you have to add more, relevance, etc. So there is a trade-off here to be achieved. Our methodology is based on training a reinforcement learning agent that uses as rewards the properties of scientific explanation. For this work, we focused on fidelity, so the accuracy of the result, but also on the relevance of the explanation to allow us to distinguish between paths that are relevant scientifically and those that are not. We measure relevance using the information content of the nodes in the graph. And this is a very simple idea that nodes that are very well connected in the graph are too general and therefore perhaps less relevant. Many things are connected to disease, many things are connected to drug, which makes those nodes less interesting to participate in an explanation. And so we train our reinforcement learning agent to be able to find paths that fit these properties. And remember, we want performance without sacrificing explainability. So above the line are non-explainable methods, below the line are explainable methods, and ours is REX. So REX achieves explanations that are better or in equal performance to other systems with explanations. And we evaluated the quality of these explanations with scientists, and this revealed that scientists prefer explanations that include multiple perspectives and that are anchored in an ontology. So they prefer more complete explanations and explanations that give you those entities as part of the explanation. But they not just only prefer those explanations, they trust the systems that are able to produce them more. So here we have the three explainable systems and a variant of our system that doesn't have the ontology part, which is the Rex light. And as you can see, the trust rating increases with completeness, causality, and relevance. So to finish, challenges for you to think about. First one, science is in constant flux. Ideas change. We used to think Lamarck's ideas on inherited characteristics were silly, but we have an increasing amount of studies in epigenetics demonstrating that there is something to be said about this. Challenge two, ontologies model a consensus, not reality. People like me who work with ontologies and knowledge graphs tend to take ontologies at face value as this truth model, but they are not. They are a consensus generated by groups of scientists. Different ontologies that model kind of the same domain very much can disagree on very basic ways to model a domain, and so tread carefully. Here we have an example, two ontologies, and in one ontology, a cell surface is part of cell part, and in the other ontology, cell parts and anatomical surfaces are disjoint concepts. They cannot be of the same kind. And this makes for a lot of mistakes when interpreting data. But even when there are well-established consensus, they change. So this is a very interesting paper. This paper took the same data set and analyzed it with different versions of the gene ontology. And the conclusions were that depending on the version of the gene ontology, the conclusions were completely different from the same data set. So as knowledge evolves, reinterpreting our old data also changes. Which brings us to the challenge of how can we tread this normal science, science that is building on pre-established knowledge, and the scientific revolutions, the paradigm changes of Thomas Kuhn. And challenge three, okay, this is one of my favorite. Most scientific data is positive. This is a challenge, a big challenge. Only 15% of published scientific results are negative. So that means that a lot of interesting data is hidden, lying in computers, in servers, and we never see the light of day. You know why? It's much harder to publish a negative result, right? But the estimates from studies that are pre-registered, like clinical studies for approval, show that out of the 94% of published studies on antidepressant were positive, but only 51% of the registered studies yielded positive results, which means most of the negative results never were published. And that creates immense bias, especially when you're training machine learning algorithms on this data. Because it means that in most real-world data sets, negative and unknown are indistinguishable. But negative results are crucial for scientific machine learning. We need them to train the algorithms. And a number of different works from protein function prediction to molecular design have shown the need and the relevance for this. And challenge 5, the last one, for which I have no slide. Scientists are people. Let's not forget that. When we design AI for science, we need to design AI for scientists. Never forget that. So the question that I finish with is, how can we support AI-based scientific discoveries that are both scientifically meaningful, grounded in prior knowledge, but not entirely shackled by existing theories? How can we strike this balance between what we know, and the new things, breaking out of the pattern, discovering new things. And just before we finish, I could not finish today and this talk, and now I'm going to be a little bit emotional. Because before being a computer scientist, I was a biologist, and this was a very inspiring person. I'm sorry. For those of you who don't know who this is Jane Goodall. Perhaps the first woman scientist that really inspired me to become one, who challenged long-established theories in the fields of both primate and human behavior. And she did so by building on existing knowledge, but breaking away from pre-established molds, both in science and as a woman scientist. And it is an honor to have been invited through the program in Women in Bioinformatics. I don't think that Jane was very much of a computer enthusiast, but she was an inspiration, and I hope that she continues to inspire future generations of women in science. Thank you. Picture if you want to check my email, because we are hiring. So we're always hiring, we are hiring, so this is the slide where you take a picture. And last but not least, the acknowledgements of everybody who funded this work, everybody who collaborated with me on this work, and also the funding institutions that allow me to be here today. Thank you so much. Questions?
So, Afonso, you wanted to?
I'm still stuck on the question of functional similarity with respect to protein interactions.
I'm sorry, the question is?
On the question of,
Yes.
I don't know exactly. It is true that proteins that, that are interacting has to some extent similar functions.
But we,
know so many counterexamples.
But we wanted to understand specifically at which level the similarity has an impact on the prediction. So what are the aspects that by being similar can influence that prediction? Doesn't mean that there aren't many other aspects playing a role here. I think that's why we reframe the question in that sense. Or maybe I'm not understanding your question.
No, I'm just
yeah.
I'm just kind of confused by the question. Obviously, this depends on the level in which you define function.
For sure.
In a sense, this is a very general level of definition of function. It's sort of a functional function, or something like that. If you think in a molecular interaction, things will become very different, for example.
Oh, for sure, for sure. So,
you define function. And obviously then you can, I don't know, I think it's a very,
so
you put it as a simple question, but I don't think that this is a simple question.
I don't think it is a simple question. It is an example of defining at which level you want to do the representation impacts how you measure similarity, which in turn impacts how that similarity can or not explain part of the interaction. I think that was what we were going for here. In more recent work, we have been integrating sequence, structure, and functional representations in embeddings with protein networks to build a more holistic representation of the process. And what we have seen in that, although I'm not presenting it here today because it is not explainable, and that's not part of the talk. Yes, I also do things that are not explainable, I'm sorry. And what we have seen there is that although sequence and structure and network topology are able to achieve good predictive performance, if you add the function-based embeddings, the embeddings based on the function for those proteins that have them, the performance of the model increases still considerably. It is not necessary to achieve good performance, but it elucidates. And the hypothesis is that there are many aspects that are important to protein interactions that are not captured by sequence or structure alone. So, you know, where the proteins are in a cell, the processes that they are involved in, the context of that interaction, which the embeddings that are based in the knowledge graph can capture and can bring that information in. So in terms of incorporating prior knowledge, that's what we are not discovering, but investigating. In terms of the explanation, for that work, we still haven't figured out a good way to ask a question that can be explainable.
And this requires a longer discussion. I'm just curious about. I see like two different ways of thinking about that. What you think from the functions to the interactions, but this is probably not the way you think has happened. The interactions were detected experimentally, and then functions were assigned later to that. So that refers only to one part of the spectrum.
Yes, yeah, definitely, definitely.
What is the top-down or
Yeah.
the bottom-up view has different consequences. And at the same time, this has to do with what we understand, the derivation that by an explanation. If you want an explanation that is good for cell biology, then
Oh, yeah.
fine. Then
Yeah,
it's
yeah,
fine.
definitely, definitely.
If you want an explanation
Yeah.
that is good for computational chemist, I will not do it.
Completely
So
different level of explanation, yes.
The intersection between going top-down, bottom-up, and the level of explainability.
So yeah, in other work, we have been actually, although we have been actually researching the different types of explanations that different experts expect for the same phenomena. So in another work we've been doing on the drug repurposing task, we have identified different explanatory needs for different experts, and we've been using the reinforcement learning agent to actually learn different types of explanations that focus more on specific types of properties to adapt to each, but again, it is still limited by what is in the knowledge graph, right? Because since we're generating our explanations based on the knowledge graph, if it's not there, we cannot capture it. But if you have a, comprehensive knowledge graph, you can generate really different explanations for the same phenomena for different experts. And that is definitely one of the things that we've been looking into. The protein-protein interaction examples are perhaps a first step towards that. And now we are asking harder questions in a sense. Yeah. And Martin had a question?
Yeah, so thanks a lot for the talk. It was very nice. I liked it a lot. I do have like two questions. One is more like on your example on different versions of gene ontology. So I'm a little puzzled that if you have quite different results, how valid is it to use this in the very first place? Because then, it might be just you have implicit bias or incomplete knowledge, or there's something implicitly in the ontologies or the extraction of these annotations. So if you're trying to exploit this approach and then you see with different versions your conclusions are very different, I'm a little worried that using the approach overall might not be, how can you assure that you have you cannot see the right answer, but a useful answer? And my second question is more like on in this presentation, we're focusing a lot on more like molecular information, experimental data. But if you go more into clinical or biomedical, medical application, there's quite a lot of bias in terms of the ontologies themselves in being in English.
Yeah.
and capturing information, specifically symptoms and more like disease-related informations or social determinants of health. We did some experiments with multilingual scenarios, Chinese and Spanish and English, and there are many clinical characteristics, descriptions, or features which you cannot even translate. And these ontologies and vocabularies, they are basically most of them, almost all of them, only like monolingual. So I'm a little worried about like our picture of health is very biased towards the representation of concepts and entities using English, American like ways of describing and seeing health and science. And the second part is like on a general perspective, like if you see this different or discrepancies, how valid is it to do it in the very first place?
So the first part, the existential problems about the gene ontology. I share them too. So that was not my paper. That was just a paper that I find that challenges something that we have as a community been taking a little bit for granted and using the gene ontology many times as a validation of many of our experimental results. I think that they found instances where the conclusions changed, but the conclusions were what are the most important functions of these gene sets, and at the top there were different functions than before. And so I would say that it changes the ranking, it might change the interpretation. It doesn't necessarily completely undermine the previous results, but they are different, and the interpretation can be different depending on who writes the analysis based on that. I think that we have here a challenge. The challenge is, are we going to believe that whatever is the latest version is the most correct version? And as you know, it's not, right? In some parts it may be, and in other parts some error may be introduced, something that later experiments, later knowledge would demonstrate is false is there. So it is always kind of the best effort. The challenge here maybe is that it would probably make sense to start to see the level of agreement with different versions when we do analysis. So is this something that, in the light of the knowledge today encoded in the gene ontology and 2 years ago, would the results be the same? Do we need to start doing this consensus or not? I don't know, but I find that we do tend to think of these knowledge repositories and sources as a single source of truth, but there's still, mistakes, biases, revisions, and everything, and we just need to take everything with a grain of salt. So I don't think there's an easy solution for that. As for the problem of multilinguality, it is a huge problem, not just in I would say not just in not having the terms in the ontologies and in the vocabularies, but also having very particular regional terms being used in clinical notes. So in Portugal, there are some symptoms that are described differently in clinical notes, whether the doctor is in Lisbon, in Porto, or in the Azores. And that also brings additional challenges into the process. I would say that Europe is very well poised to break out of the English only or English first challenges in medical knowledge, but there is a very strong bias introduced also by scientific publications. Because if you are doing systems that include not just the clinical notes and the EHR, but also scientific publications, and those are going to all be in English, you will still have a strong bias there. And I don't know exactly how to break out of that bias. I don't know if translation models are sufficient to answer those needs. Do you think they are?
Soon.
Let's hope so. Okay, any more questions? Okay, should we finish? Okay, you can ask your question as I walk away. Okay. No. Okay. Okay. So quick question.
I'm not sure
I'll
what's
try not to take
going to
too
be an
long.
interesting question because I'm neither biologist or computer scientist, so it's probably more my misunderstanding. But
So what are you?
I don't know. Lots.
That's a good answer.
No, it's because is it like it's just the explanation part of all this model is more? Is it more like the direction you take is like more like asking a model to answer more specific question rather than having better explainability about how the model works? Because so I understand like initially it was like similarity between protein, that's it. And now it's like more complex question. And then the model answers just more complex question, but it's just the explainability happens before and after the model, but not within the model itself.
I would say that in the counterfactual model, it happens within the model because you train the model to represent the proteins with the different shared functions, okay? And then you do a perturbation in the model. So, but it's still the same model that you're still running the same model to generate the prediction and the explanation. You're just giving it more or less data, okay, to ask the counterfactual question. So the explanation is still generated. It's within the model in that sense. And in the pathfinding explanation, the explanation with the reinforcement learning agent that learns the path, the path is the answer to the prediction. So the goal is to reach the reach the disease starting from the drug, and the path that is chosen to reach the disease is both the prediction, this is the disease, but also the explanation because you have the path, right? So it's part of the model as well. Okay, you are a wonderful audience. Thank you very much. And I need to take a picture, and I would like all of the ladies in the room to join me for the picture, for the bioinformatics for women thing. I'm sorry, Alfonso, for taking this a little bit longer.